Hello, guys. Good morning. Good morning, everyone. How are you all doing? Good morning. So, how are you all doing? Yesterday, how was the session? Did you enjoy it? So, yesterday we left with certain two questions, right, related to GPCR. So, did you all solve those questions? Because we'll be doing it today. Good morning, Mahima. Okay, it was great. So did you go back and you went through your books, like standard books, because those everything I explained from the standard books, Lodish and Leringer. So it would be easier for you, like after yesterday's session, understanding all the signaling processes in GPCR, it would be easier for you now, right? Hi, Lakshman. Hi, Sunita. Good morning. Good morning, Devanshi. Yes, Mahima, we'll be discussing that. Okay. Hello, Millennial Chakravarti. So today I have come up with Unit 5. So in Unit 5, we'll be discussing about some basic terms that are being used. Like these terms are very much important if you need to understand the whole developmental biology. Right, so these basic terms, understanding the differences between these terms, because these terms are very much confusing. How development in an animal actually starts. So we'll be beginning from like from the start. We'll be starting and we'll be ending it, like where it ends, like how senescence takes place and how an organism actually develops, what all processes it goes through, what all signaling processes, like it goes through which, like it goes from a developmental stage, right, from the young ones to the adult ones. So all these things we'll be discussing in developmental biology, and it's going to be very interesting session today, right, because it's actually related to us only, how we developed. So yes, one more information regarding like today is that today there would be a live session at 4 o'clock where the experts, sorry, at 5 o'clock it would be, where the experts, they will be discussing how the paper was, and on 17th, the CSIR NET paper, the difficulty level of the paper they will be discussing. So general idea you'll get, and like unit wise we'll be discussing. Experts, they'll be telling you like what all things you need to be like needed to be done at that particular point of time, and what was the difficulty level of the exam, and everything would be discussed in that particular session. So 4 o'clock, stay and watch that particular video, so you'll get an idea regarding like how you actually did your paper, how the paper was really set up. So next, for the next paper, you get mentally ready, right? So we'll be starting, and the topic of the discussion would be from Unit 5, the basics of development. So here we'll be discussing about the key events that take place in the development process. Then there will be the what is the potency and what is the cell fate, what is the cell lineage. We'll be understand these terms, and we'll be knowing about the types of potencies. Then we'll be coming to the simplified development hierarchy in vertebrates. So what is the hierarchy in which there is like ectoderm, mesoderm, and endoderm, right? The three germ layers are there. So which germ layer produces which kind of organs or tissues, that we'll be discussing. Then comes the comparison between the different mechanisms of developmental patterning, right? Then there will be comparison between types of specifications would be there. So in like mechanisms of development patterning, we'll be studying about the concept of specification, determination, differentiation, all these things we'll be discussing. right? And then types of specification. What are different types of specification, such as autonomous? What is synthetal specification? And what is conditional specification? Like, what is the difference between all of these three? Then coming to the comparison between the morphogen and cytoplasmic determinants. So there will be we come across these terms like what are morphogens and what are cytoplasmic difference. Like most of the time we get confused between these terms. So what is the difference between both of these terms and where it is actually being used? All these things will be studying. Then we'll be coming to the comparison between the induction and competence, right? What are induced cells, what are responding cells, and how a responding cell is competent to particular induced signal that is being produced by the inducer, right? Then we'll be comparing between the modes of inductive instructions that we have, right? So this is the overview of the content for today's session. So yesterday we left at these two questions were being asked, right? So many of you would have solved it, right? Those of you who attended the session, and those of you didn't attend the session and you're new here, they can also see the question and think for themselves like what would be the right answer for these questions, right? So I would be expecting answers. So first question is that the glucose is mobilized in muscle when epinephrine activates GS alpha. In an experiment in which the muscle cells were stimulated with epinephrine, it was being seen that the glucose mobilization was observed even after the withdrawal of the epinephrine. This could be. So what would be the right option for this? Like even after even after the epinephrine was withdrawn, still this GPCR signaling was taking place. So what would be the correct option for this? Anybody? Okay, so Aubrey says option A. Then any other answers? Okay, due to the presence of protein kinase A. Okay, Mahima says due to the presence of protein kinase A, right? Okay, video zone, he says C option due to the presence of CMP phosphodiester activator. Okay. Devanshi Soni says A. Amala says C. So different options are coming for this. So yeah. So this question basically says that even after the withdrawal of epinephrine, we see that still there is the transduction of the signaling, GPCR signaling is taking place, right? This is basically due to the option A is the correct one here, because due to the presence of CAMP phosphodiesterase inhibitor, because CAMP phosphodiesterase is an enzyme, as we have discussed yesterday. So as we've discussed yesterday, the PDE basically is the PDE, that is the phosphodiesterase that we short form was the PDE. So this PDE was basically responsible for converting the cyclic CMP into the normal form of CMP, right? The 5 CMP. So wait a second, my pen is not working. Okay, I'll explain it like that only. So here we said that I'll change the color. Yes. So here, as we have, as I have explained yesterday, that PDE, that is the phosphodiesterase enzyme, it is basically responsible for converting the cyclic AMP into the normal form of CMP, right? A normal form of that is the 5' AMP. It converts the CAMP into the 5' AMP, right? And this is when the concentration of the CMP, it gets reduced inside the cell. As a result, the GPCR, that signaling, it stops. But if there is the presence of the inhibitor, the CAMP phosphodiesterase inhibitor is present, it will inhibit this particular enzyme. As a result, the concentration of CMP will still be high in cytoplasm. As a result, the GPCR signaling will still be continuing, right? So option A would be the correct one for this. So those of you who marked option A are right? Then coming to the second question. So second question says that in dark, rods show a large inward dark current which is suppressed by a flash of light. Which one of the following statements explaining the effect of the light is true? So first option is the sodium channels in the outer segment of the rods are closed. Second is the cytoplasmic CGMP concentration increases. Third is the sodium channels in the inner segment of the rods are closed. And last option is transducin dissociated from the beta arrestin. So what would be the correct option for this particular question? Option C. Okay. Yeah. So option C is the correct one, because what happens is when the light, like the opsin protein, it absorbs the light in case of rhodopsin, then what happens, that CGMP, it falls off, and the concentration of CGMP in the cytoplasm, it decreases. And as a result, the channel for the sodium channels, it gets closed, right? And it only happens in the inner segment of the rods, not the outer segment, right? So those of you who gave the C option are the correct one, right? So yeah, right. So let's begin. So here is the first slide coming on your screen. So here we'll be discussing about the key events in the animal development, right? So there were basically two theories which were being given during. So firstly, I would want to ask you, what is the difference between development, proliferation, and growth? Like, these are the three terms we often confuse between them, and these are the individual terms. So you need to tell what is the difference between all of these three terms, because development is not equal to growth, and like proliferation is also something very different. So can anybody tell me what is the difference between development, growth, and proliferation? Yeah, so I would be expecting answers from you. This is the general thing regarding development. Right. Kaveri says, ma'am, why not answer D? Okay, in question 1. Due to like protein kinase A will be there, like even if the GPCR like it is activating, and even if GPCR is not activating, still protein kinase A would be there. But protein kinase A will only be activated when this concentration of CMP would be higher in the cytoplasm. Unless and until the concentration like of the CMP is like not high, then this protein kinase A won't be working, right? So were you able to relate, Kaveri? Did you get it? That option D is not the correct one. Option A is the correct one. So yeah. So development is a process which we which we go through, or which we see in like all the stages of our life. Like at this particular point of time also we are developing. When we are younger, we were that time also we were developing, and when we are old, that at that particular point of time also we are developing. So at any age, the development process, it continues. But growth is something which happens till a particular like age. It doesn't happen throughout the life, right? So in growth, basically what happens, the size basically increases. And in development, it is basically related to the maturity, how mature a particular person is becoming. So that is something which is telling about the development of a particular kind of individual, right? Then comes the proliferation. So proliferation is basically the increase in number, right? Growth and proliferation are not same. Growth, just size gets increased, and in proliferation the number gets increased, right? And development and growth is also not the same thing, because development is directly related to like maturity, and growth is basically related to the increase in size, and it doesn't happen like throughout the age. So as you would have seen, like when we study cell division, right? So we have how many stages? Like there is the gap 1 stage, right? Then there is the S phase, then comes the G2 phase, then comes the M phase. So G1 phase is the phase when the cell is basically, it is producing certain kind of proteins and all those factors which are responsible for the cell to divide. So at G1 phase, the cell is active. But what happens, the number of the cells, it doesn't get increased. The cell is still in the developmental stage. The cell is developing, but there is no proliferation which is taking place, right? Proliferation is not taking place. So this is the basic difference between these three terms. Development is and like proliferation and the growth. All these three terms are very much different and like separate from each other, right? So key events in animal development. So there were two theories which were being given regarding development. First one was the old theory that is known as the pre-formation theory. So here what happens, it was being said, there was a scientist who said that you can see that the diagram is being given here, right? So there is a sperm here. Inside it, you can see there is a tiny person there, right? So according to the pre-formation theory, the whatever things that are required for us to develop into an organism, it is already present in this inside the sperm in the tiny form, right? And then what happens when the growth happens and everything happens, then this tiny little person, it gets developed into like in a full organism like us. But there was a major drawback regarding this particular theory, the pre-formation theory, that everything is already formed inside the sperm. So if everything is already formed inside the cell, then these and the inside the sperms, and so everyone should look identical to each other, right? But what happens are there is like two types, like XX is there and XY is there, right? We develop into like females and males. So this shouldn't be the case if like everything is pre-formed, everything is decided inside the sperm itself. So two different individuals wouldn't have been formed, two different genders wouldn't have been there if this theory was correct. So this was the major drawback of this particular theory, pre-formation theory, which says that everything is pre-formed inside the sperm, and when the organization and development takes place, then what happens, this particular sperm gets converted into an organism. And this was the incorrect theory. After this came the theory of epigenesis, right? So this theory was being considered as the right theory because, so epigenesis basically means the gradual development of an individual. So if we talk about a flower, so a flower, like it starts from a bud. So the bud, it grows, and it then slowly and steadily it gets developed into a flower, right? It doesn't happen like in one day. One day a bud doesn't gets converted into a flower. So it is a slow and steady process. It is being said development is a slow and steady process. It doesn't happen overnight. So here you can see that in epigenetics, there is a diagram. It is being shown here. So in epigenetics, we study about the bidirectional influence, right? So majorly the development, the things that are required are genetic activity. So firstly, in our genes itself, like from DNA, you would have read the central dogma. From DNA, replication takes place. After replication, transcription takes place, and mRNA is being formed. And from this mRNA, the proteins are being formed, right? These proteins express themselves, and the functions are being conducted inside it, and an organism, it develops from the younger one to the adult one. So genetics plays a very major role. right? Then comes the neural activity. So if you would consider a twin, like, so these twins are basically, they are identical to each other from head to toe, right? But still they have different behavioral differences in them, right? One person behaves differently and the other person behaves differently, even if they look very much similar to each other, right? On that basis, you can differentiate between the two twins. So neural activity, even if the person, like two people are very much same, the neural activity is different in them, right? The behavior is different in them. The neural, through neural activity only the behavior of the particular organism is being decided, right? And this behavior change is basically being controlled by the environment, in which kind of environment a person is basically living, or an organism is basically living. So everything is interlinked here. You can see that that environment, it directly controls the genetics of an organism, right? So in epigenetics, this particular theory is considered to be far more justified for the development. So epigenetics was the second theory, and it was being considered correct, right? And the first theory, that was the old theory, that is the pre-formation theory, it was being considered incorrect. After this, what happens? That we know what are the different stages of the development. So here is an amphibian frog that I have taken, right? So what happens that these this amphibian, this xenopass, it basically produces gametes, right? So first stage is gametogenesis. So gametogenesis takes place, sperm and eggs are being produced. These sperm eggs, they fuse with each other and they fertilize. So fertilization takes place, right? Second is the fertilization. After fertilization, what happens? A zygote is being formed. This zygote goes through many cleavage steps, right? So this fertilized eggs, it gets cleaved. And can you please tell me, like, which is the first stage that a particular fertilized egg, when it gets cleaved, what is the first stage it is being it forms, and how many like cells are being formed in that particular stage? Yeah, so Maima says the development is continuous. It is throughout, like, yeah, in and whereas the growth is related to the enhancement in size, right, into a certain time interval. Yes, Maima, that's what I explained. That growth, development, and proliferation are three different terms. So yeah, second, the question that I asked was that there is a fertilized egg, a zygote, and what happens is when this particular zygote, it gets cleaved, it forms a cell, like a ball of structure, which has a particular name, and it consists of certain number of cells. So you need to tell what is the name of this cell and how many cells are being produced in this particular structure. Yeah, so Kaveri said it's a Morella stage and 16. Yeah, Kaveri, you're right. So first stage is the, like, division takes place 4 to 16. So 16 cell stage is basically the Morella stage, which is being formed, right? So as we know that in cell division, how many phases are there? G1 phase, M phase, S phase, and G2 phase. So when a fertilized egg, it divides and it converts into like blastula, all these things are going on. When the cleavage basically takes place at this particular point of time, this G1 phase is not required there, right? Neither the G1 nor the G2 phase, because these two phases are basically the preparatory phase. Like at this particular point of time, what happens is there are certain proteins that are being formed which prepares the cell for the next upcoming phase, right? But as we know that the egg already have all those components which are required for a cell to divide. So the cell doesn't need the particular phase or time for producing those proteins which are required for development, because those proteins and those components and enzymes, they're already present inside the egg, right? So G1 and G2 phase doesn't take place. Only M and S phase are present in when the fertilized egg, it gets like divided and gets cleaved and forms morel and all these stuffs, when this proliferation basically takes place. That's why inside the zygote, the proliferation step, it takes place very fast, right? So this much have everyone understood. Yeah, Yes, Jagdish, it is the Morella stage, right? So after Morella, what comes is the Blastula stage. So Blastula is basically the 32 cell stage, 32 to 64, you can say, right? So in Blastula, what happens is you can see that there is a cavity which is being formed, right? A cavity is being formed, and cells, they align themselves at the wall of this particular cell, right? Then comes the gastrulation. Gastrulation is a step in which basically what happens, three germ layers are being formed. The three germ layers are basically ectoderm, mesoderm, and endoderm, right? So these three germ layers are being formed in gastrulation. Then comes organogenesis. In organogenesis, what happens, from gastrula stage, a neurular stage is being formed. So organs inside the body of a particular organism, it starts forming, right? Then hatching take place. Tadpole larva, it gets converted, and this process is basically known as the metamorphism. So metamorphosis process takes place. This is the process which is basically lies during the transition from the from the gastrula stage to the adult stage. So to form the adult from the organogenesis stage, this particular metamorphosis step, it is the transition step in between which lies, right? So this is the major outline of how the stages of development basically happens, what are the events which takes place, and how things are basically being formed from a single cell to an adult, right? So here it comes that the process of development include fertilization, cleavage, gastrulation, organogenesis, metamorphosis, all these things we discussed. So you can see that at every stage the development is taking place. So you can say that the development, it happens throughout. So this is the basic definition of the development, while growth doesn't happen throughout, right? Next important term comes the potency and the cell fate. So these are very important terms. Many times the questions are being asked related to these terms in CSIR NET. So what are the basic rules related to potency and cell fate, right? So firstly, knowing what is cell fate, what is cell lineage, and what is potency. What does these terms really means? So first comes the cell fate. So cell fate is basically the differentiation into a particular cell. So a particular cell is there and it gets differentiated into a particular type of cell. So what happens is, we take Blastula, right? Let me change the color. So here it won't be visible. Yeah. So this is the Blastula that I have taken, right? This Blastula, during this Blastula will form three germ layers, right? Ectoderm, it will form, and then it will form mesoderm, and then it will form endoderm. So in ectoderm, what happens? This particular germ layer, it basically forms the nervous system, right? And nervous system will basically form the CNS, and everything will be forming here, and it will at last it will form neurons. Right. So what happens is neurons, that is from the Blastula stage to neurons, this is the fate of the cell. So from Blastula stage, the particular cell, it got committed and it formed neurons. This is the fate of the cell. This is known as a cell fate, right? And from Blastula till neurons, the whole history of the cell, this is the cell history, right? This history of the cell is basically known as the cell lineage. Okay, these two terms, did you understood that? Okay, so factual biology, he says that clarify about the controversy questions which comes in CSIR. Okay, so we will be having the webinar session today now. So in that, the experts would be coming. They'll be discussing about the difficulty level of the questions, how the question was actually being framed, right? This will give you a general overview. After that, from the next week, the experts, they will be discussing all the questions that has come in CSIR NET exam in this particular year that you have given. And there you will be knowing what would be the correct answer and how you need to be attempted that particular question. No? So all these things would be discussed in upcoming week. So it will go for a week and everything is planned and every help would be provided to all of you, right? So don't worry. So So these two terms, did you get it? Yeah. So first is the cell fate and second is the cell lineage. So from the blastula till neurons, it is the history of the cells which basically defines the cell lineage term, right? So cell lineage, it says that the developmental history of a cell from birth until its final division. So whole history of the cell from the blastula stage till the formation of the neurons is known as the cell lineage, while the fate of the cell is that it differentiates into a particular cell type. So a cell gets committed and it differentiates into a particular cell type, such as the ectoderm cell, it got committed to form the neural cells. So this is the particular fate of the cell, right? Then comes the potency. Now potency is something, it is the intrinsic property of a cell, right? And it basically, what is the potential of the cell? Which kind of cell, like from a cell, wait a second. Yeah. So potency basically is the intrinsic property of the cell, right? And it basically tells that the cell fate is equal to the potency plus the environment. So the fate of the cell is basically equal to, or it could be, yeah, it is basically equal to the potency. Potency plus the environment. So environmental factor also plays a role here, right? So potency is basically like how a particular cell, or how what is the capacity of the cell to get converted into a different kind of a cell, right? So there are basically 4 types of potencies in which it is being measured. There could be the totipotent cell, there could be the pluripotent cell, there could be the multipotent cell and unipotent cells. So the cell, when it is present in its zygotic form, like fertilized cell, it is known as the totipotent cell because it is capable of forming any type of cell or any type of organ, or like it is capable of forming a whole organism actually. So totipotent cells are those kind of cells. So potency basically tells like what is the potential of a particular cell to get converted into which type of cell, right? And pluripotency is basically that there is a limited, like you can see that in ECS is basically the embryonic stem cells, right? So embryonic stem cells, what happens, these embryonic stem cells, they are considered as the pluripotent cells because they are capable of forming the like any organism it can form. But what happens, only the wait a second. So totipotency, you understood, right? Totipotent cells are basically the fertilized eggs which are capable of forming a whole organism. Pluripotent cells, what happens, they have the limited capacity to form a particular kind of cells only. It cannot form the entire organism, right? And multipotent cells also, they also have the yet another like limited amount that they can form, such as the if I say about the blood cells, the hematopoietic cells, they are able to form only the blood cells. So these are considered into the multipotency. And unipotency is that the neural cells, they are only able to form the neurons. So this comes, the restriction comes here, right? So these are the 4 different types of potencies in which like cells are being divided into. So in upcoming slides, you'll get more clarity about what potency actually means and how these different terms are basically being like different from each other, right? So from this concept wheel, you can understand that cell potency is always greater or equal to the fate. So whatever the fate of the cell would be, the potency or the potential of the cell would always be greater than it or equal to it. It can never be less than the cell fate, right? So as we discussed, from the blastula, the neurons were being formed, right? So these neurons, they had a particular, like the cell, it got committed and it had a particular fate for itself. It formed the neurons. But the whole potential of a particular cell from the blastula stage till the fate of the cell, its potential was like huge. It could have formed any kind of cell, but when it got committed, then only it formed the particular type of neurons only, right? So the potential of the cell is very much high. So it is always greater or it is equal to the cell fate. Then comes, you already know that potency is the intrinsic property of a particular cell. So it would be present inside the cell. The cell only it will decide like to which kind of particular cell type it will be formed, right? Then comes the cell fate is equal to the potency plus the environment. So cell fate, it majorly depends upon the potency or the capability of the cell to differentiate into a particular type of cell type, and it is also dependent upon the environment it is being kept. So if what happens is, if we keep a particular kind of cell into a like there is a neuron cell, if we have kept it into the muscle cells and we have provided all the the factors that are being required for this particular kind of cell to get converted into the muscle cell, it will happen that it will get converted into the muscle cell because we have provided all the kind of factors, right? So on the basis of the external environment also, the fate of the cell is being decided. So then coming to the cell fate becomes increasingly restricted until a cell is terminally differentiated, right? So the fate of the cell, it becomes increasingly restricted. So when the cell, it continues and it grows and gets committed, then it gets determined and then it gets differentiated. So at differentiation level, what happens? The restriction comes here. So a particular cell, it will be only be able to form a particular kind of cell only. It won't be able to form another kind of cell. So coming to an example, what happens is when we grew up, so till 12th, what happened? We had many options. We decided that we can become doctor, or we can become engineer, or we can become a singer, or anything, right? So we had like choices were there. So then what happened? We did our graduation, we did then our masters. Then what happened? At a particular specialization only, we gained knowledge and we became committed to that particular field, right? Then we are giving here CSIR NET paper, and we are pursuing like for PhD we are going. So this comes, restriction comes here. Now we cannot change our field, right? So this is what happens for the cell also. Once they are committed, once they are determined to form a particular type of cells, they cannot revert back, right? So there comes the restriction here. Then comes the cell fate and the potency are progressively restricted. So when the progressive development take place, right, then the cell fate and the potential of the cell, it gets restricted. It can only produce a certain type of cell and it cannot revert back, right? So this is what this concept wheel is basically telling. So now have you understood like what potency actually means and what is the cell fate? Yes, committed stage is the determined stage, right? Yeah, yes, yes, Mahima. Hematopoietic cells are basically the multipotent cells. So this much have you got clear with, or should do you want me to repeat it? Then we'll move forward. Then comes to the different stages of potency. The types of potencies are there. So first is the totipotency, then comes the pluripotency, then comes the multipotent cells, then comes the oligopotent and the unipotent. So from the diagram here, first I'll try to explain you. So totipotent cells basically are present in the zygotes, right? So there is a zygote. The cells inside the zygote would be totipotent. They would be capable of producing an entire organism, right? Then comes the pluripotency. So the embryonic stem cells, or the PGC cells. So embryonic stem cells, they are basically like capable. They are capable of producing. They are not able to produce the whole organism. Like they are able to produce any type of cells, but but they are not able to capable of producing the whole organism. There comes a restriction there. So then comes the multipotency. So in case of multipotency, what happens is when this particular cell having the inner cell mass. So this inner cell mass is basically the thing which consists of the pluripotency in them, right? These are pluripotent. So when the cell gets further differentiation takes place, and this cell gets into a stage where then the cells are basically multipotent. So Gastrula stage comes there. So in Gastrula stage, what happens is the three germ layers are being formed. The ectoderm is being formed, the endoderm is being formed, and the mesoderm is being formed, right? So ectoderm will only be producing the neural stem cells. So they like multiple neural cells, stem cells they will be producing. And endoderm will be producing a certain other kind of cells, epithelial stem cells only. And mesoderm germ layer, it will be producing the hematopoietic stem cells only, right? So what happens is here the restriction comes. So multipotent cells are the one which do not have the capability to produce the whole organism, right? Only a particular type of cells only they can produce. Then comes the unipotency. Unipotency, what happens when these neural stem cells, it further gets differentiated, it can only be producing the neurons, right? Epithelial cells, stem cells, they will be producing the gut cell, and the hematopoietic stem cells, they will be producing the blood cells, right? So this is how it happens. So from the this concept wheel, we can understand that totipotent basically means the omnipotent. That is, it can form the whole organism. It is present everywhere. Omnipotent basically means that. So stem cells that can differentiate into an entire organism results from the fusion of the egg and sperm. So from the fusion of the egg and sperm, the zygote is being formed. This the cells in the zygote, these are basically the totipotent cells, right? Then comes the pluripotency. So stem cells that can differentiate into any tissues type except for the placenta tissue, right? So only the placenta tissues they cannot differentiate into. Other than that, all different kind of tissues they can differentiate. So here comes the pluripotent cells. So extra embryonic part it cannot produce, but other parts it can produce, right? So the pluripotency, it has two major properties in it. First property is the self-renewal property. It can self-renew itself, right? It can make its multiple copies. Apart from that, it can differentiate into other cell types also. These are the two properties of the pluripotent cells. While the multipotent cells, it is not capable of self-renewable. It cannot renew itself, right? Adult stem cells are basically the multipotent cells. It basically like have the property of multipotency. Then comes the multipotent. So multipotent stem cells that can differentiate into the multiple cells in a closely related family of cells. So closely related family of cells only it can produce. It can not differentiate into some other family type, right? So here it is being explained that the neural stem cells, it will only form the neurons, right? It cannot form the gut cells. So then coming to the unipotent. So unipotent, what happens? The stem cells that can differentiate into only one cell type, but still possess the self-renewable, right? So what happens in unipotency? They form a particular type of cell only. They cannot produce some other type of cell, but they have the self-renewal property. They can produce their own types. They'll be able to produce their own types, but they cannot produce some other type of cells, right? This is the basic definition of unipotency. So from this diagram, you would have get an idea about like how totipotent cells, what are the examples of totipotent cells, and how they are different from like pluripotency and multipotency and unipotency, and what are the different examples of all these potencies, right? So here comes the simplified developmental hierarchy in vertebrate. So as we have discussed, like a particular organism is there, fertilization takes place, egg and sperm it fuses, produces a zygote. Then what happens? A blastula stage comes firstly morula is being formed, a 16 cell stage. Then 64 cell stage is being formed, that is the blastula. And after that the gastrulation takes place. So in gastrulation, what happens? The 3 germ layers are being formed. So here is the particular flow chart. In the form flow chart, I have explained what type of cells ectoderms form, what type of cells mesoderm form, and what type of cells endoderm or the internal layer forms, right? So ectoderm will be forming the outer surface. It will be forming the epidermal cells of the skin. Secondly, it is also capable of forming the neuron of the brain, right? The central nervous system part will be forming. And other than that, it can also form the neural crest. Then comes the mesoderm. In mesoderm, what happens? the dorsal part is being formed, notochord is being formed, the paraxial part, that is the bone tissues are being formed, and intermediate part, that is the tubule cells of the kidney is being formed. And the red blood cells also, these are also being formed in the by the mesoderm germ layer. So hematopoiesis is the event that basically takes place through which the red blood cells are being formed, right? Then comes the endoderm. In endoderm, what happens? The digestive tube, that is the stomach cells, are being formed here. So all these are basically the cell fate. So right from the Gastrula stage till the stage where the particular cell type is being formed, this is basically the cell fate, right? This is deciding the fate of the cell. So cell got committed to produce a certain kind of cell, right? A certain type of cell. Then what happens? It gets into the determination stage. After that, it gets differentiated into a particular type of cell. So here comes the cell fate. Now, from here, from Gastrula till this cell fate, this is basically telling the history of the cell, right? This is known as the cell lineage. So in this chart form, you can understand like what is the cell lineage and what in which step the cells is basically being forming from in ectoderm, which kind of cells is being forming, in mesoderm, what kind of cells is forming, and in endoderm, what kind of cells inside a body is being forming, right? Then at last comes the germ cells. The germ cells are basically in males it is sperms, and in female it is egg, right? So here is the reminder for all of you that Shorter batch would be starting from 4th of March, and it would be targeting the CSIR NET 2022, and the rate classes it will here we'll be giving you facilities, and it would be consisting of the rate classes by CSIR NET experts. Then there will be interactive online classes. So 24/7 your doubts will be cleared through chats, and study materials will also be provided to you, right? So if you want to know more about the Shorter batch and what all things or facilities that is being provided, you can mail us. The team will guide you, and you can also call us on the toll-free number that is flashing on your screen, right? So yeah, so here comes the major question: how a cell develops into an organism, right? To answer this particular question, we need to answer these 3 sub-questions are there. So first is what force affects the development of an embryo. Second is how does the cell differentiate via influence of these forces. And third is how does these cells organize themselves into tissue and organs, right? We'll be answering and getting an answer to all of these questions in the upcoming slides. So first question says, what force affects the development of embryo? So in that, for the development, there are basically 2 factors which are responsible for the development of an organism. First is the external factor, and second is the internal factor, right? So in external factor, what happens it basically influences the development of embryo from outside. External factors could be temperature, light, humidity, predators, or competitors, right? One of the very famous example of this external factor could be the effect of temperature on crocodile eggs, right? So in crocodile, what happens is whether a male would be formed or a female would be formed, it basically depends upon at what temperature the incubation of eggs basically taking place, right? So incubation of eggs at 33 degrees Celsius, it produces mostly males. While when we lower down the temperature below 33 degrees Celsius, what happens? Females are being produced. So enzyme here, which is basically responsible for the sex determination, is the aromatase enzyme, right? This enzyme is very important. Questions related to it has come in CSIR NET paper. So you need to remember there is an enzyme. This aromatase enzyme, basically what happens is, this is basically affected by the temperature. So it, due to the effect of temperature, it basically determines into which sex a particular crocodile will be forming. So yeah, this is the basic example. Then coming to the internal factors. In internal factors, it basically says that influence the development of embryo from inside the body. So these intrinsic factors, these are majorly present inside the body of the organism itself, and internally it produces signals and everything, and after that only a particular cell responds and produces a certain type of organism. Now the example of these internal factors could be the mRNAs, right? The mRNAs or the genes basically which is responsible. So mRNA example the bicoid and nanos in case of Drosophila melanogaster. So in fruit fly, what happens is there are two types of mRNAs which are present. This is the bicoid and nanos. So these are the internal factors, right? When the concentration gradients, like these basically forms a particular type of particular part of this Drosophila melanogaster on the basis of its gradient level. What is its concentration gradient, right? So bicoid is majorly responsible for forming the anterior part, and the nanos is the mRNA, or it basically produces a protein, nanos protein, which is basically responsible for the production of the posterior part. So what happens in Drosophila? So this is the structure of the egg of the Drosophila. So what happens is the gradient form. So this bicoid mRNA is basically present at this particular part, right? It present in anterior part, and this is the posterior part. So in the anterior part, this bicoid mRNA is present. So this bicoid mRNA, when it expresses itself and produces the bicoid protein, so it's the order of its concentration is basically it is high at this particular portion in the anterior portion, and slowly it decreases, right, coming to the posterior part. So this should be the concentration gradient. So because of this, what happens, this bicoid protein, it produces the anterior part, and the mRNA, that the nanos mRNA, it is the nanos mRNA, it is basically present in the posterior part, and it produces the protein, and its concentration gradient is basically, it is present in high amount, in high concentration in the posterior part, and in less, and when it goes or travels towards the anterior part, its concentration decreases. So this is the kind of morphogenetic gradient level of both of these proteins, and on the basis of this gradient level, the particular part of which particular part of the Drosophila melanogaster would be produced, it is being decided, right? So these are basically the internal factors. It happens within the organism itself. So these two examples would be clear to all of you. So these are the two factors which are basically it affects the development of a particular organism, right? So I must say pluripotent and induced pluripotent are similar. Yes, Mahima. Induced pluripotent, we'll be discussing about that in upcoming slides, and then you'll get your answer, right? So coming to the comparison between the different mechanisms of developmental patterning, right? So here are the different mechanisms through which the development of an organism takes place. So second question that we had here was how does the cells differentiate via influence of these factors? So when these factors come into play, so how a particular cell, it gets differentiated into its fate. So for that, there comes the three major events which takes place, right? Firstly, the cell commits itself, then the determination takes place, and after that, the differentiation takes place. Now we'll be knowing what is the difference between these three terms. So here it is written that during embryogenesis, an undifferentiated cell, it matures through specific stages that cumulatively commit it to a specific fate in the following order. So first comes the specification, right? Specification and commitment both are same thing. Then comes the determination. So firstly, the cell commits itself to produce a particular kind of like cell type. Then it determines itself, and after that, it differentiates into that particular type of cell type, right? So here, through the help of this comparison table, firstly, in case of commitment, it says that before a cell overtly differentiates, it can be said to be committed. That is, the cell has been instructed on its fate. So cell is being given an instruction that it will be producing a certain type of cell. So its fate is already being decided. So cell has decided its fate, right? But it can revert back also. This is the property of commitment. Like there is the reverting property takes place. But in case of determination, it doesn't happen like that, right? So secondly, it says that in commitment, it is the intrinsic program inside the cell, right, is activated within a committed cell that causes it to follow a particular pathway of development, right? So internally, signals are being produced once the cell has decided its fate. Internally, some signals would be produced and some enzymes would get activated, proteins would get activated, there would be crosstalk taking place, and the cell would be it would be following a certain pathway to get differentiated into a particular type of cell, whatever cell fate it has decided for itself, right? So it basically depends upon the intrinsic factors also, and it also depends upon the external environment also, right? So all these intrinsic and extrinsic factors will play a role here, right? Then comes, as the cell fate becomes restricted, following each decision in the developmental hierarchy, cells become committed to a certain fate. So when the cell it has started following that particular pathway, then what happens, it becomes more committed to come to its certain cell fate, whatever cell fate it has decided. Once the pathway it has started following, it will ultimately form that cell fate, right? It has become committed to form that particular type of cell. Then it says that the fate of a cell is said to be specified if the cell is directed to follow a certain development pathway and does so when placed in isolation, which should provide a neutral environment, right? So this particular statement basically says that in this committed state also, what happens if these particular cells, they are being placed into some other cell type, right? Such as we have placed neural cells into the blood cells, right? And we have given all the factors that is responsible to convert this neural cell, which is in the committed state, to get converted into the muscle cell type or the blood cell type, whichever cell type, right? When we give these kind of factors, then what happens, these committed cells, it gets influenced by these environmental factors, these external factors actually. Those factors, it converts this cell, this committed cell, which is committed, basically it has decided that it will form the neural cells, but when it is being given the external factors which are responsible to produce the muscle cell or other type of cell, then what happens, it goes through that pathway and it gets converted into the muscle cell, right? So what happens, the reverting is there. So when the cell is in its committed state, it can also convert into some other type of cell if the external factors, it varies, right? Then it says that the commitment at this stage is reversible, as it may be specified in a different way if placed in a different environment, right? So whatever I explained, that's what this particular line is saying, that if the external environment is different, then the cell has the capability that it can get influenced by the external environment and get converted into other type of cell, right? Then it says that the two major strategies for establishing commitment is through the inheritance of the cytoplasmic determinants and the external inductive signals, right? So two major factors on which the cell, it basically strategy it establishes to form a particular cell type. Two major factors which are responsible, which basically influences a cell fate are, first is the cytoplasmic determinants, about that we'll be studying. So these are basically some kind of chemicals that are being produced or signal molecules that are being produced inside, which are basically responsible or which induces a cell to follow a particular pathway and get converted into particular type of cell, right? This is the intrinsic factor. Second is the extrinsic factor, which is the outside environment which is responsible. So if you give an external environment to a particular cell which is in its committed state, those factors, they can influence this cell, and this cell can revert back and get converted into the other type of cell, right? So both of these external intrinsic factors, they play a major role here, right? Then coming to the determination. So in determination, it says that the progressive restriction in their developmental potentials, right? So when the commitment took place, that the cell has decided that which particular cell fate it will be going through. Then what happens is the cell has determined itself that this is the only thing that I want to do. This is the only thing, the only cell that I want to get converted into, right? So at this particular stage, what happens? The restriction comes here. So the cell is restricted now. It doesn't have a choice to get converted into some other type of cell. It is restricted to a certain pathway, and it will follow that pathway, and it will produce a certain type of cell only, right? Then comes the determination to follow one developmental pathway coincides with the loss of the competence to follow the alternative pathway. So what happens is, when a cell is determined to follow a particular developmental pathway and get converted into a particular type of cell, then all the alternative pathways which are responsible to produce some other type of cell, the cell is restricted for that. It cannot go through or revert back and follow some other pathway and get converted into some other cell type. So what happens? There is a restriction which comes here. So competence, that is, the cell is not capable of those kind of signals which are being given outside. Like, even if the cell which is in its determination stage, even if we place into or give such kind of external factors to the particular cell which is determinate, that it can convert into some other type of cell, it won't do that, because it is determined to get into a particular fate, right? Then comes that the determination implies a stable change. The fate of the determined cells does not change, right? There is a stable product or the stable cell type it will be producing. It won't change its cell type. Then comes that the fate of the cell is said to be determined if it cannot be changed, right? And regardless of the cell's environment, even if we change the environment of the cell, still the cell is determined to produce a certain kind of cell type. Then comes that the commitment at this stage becomes irreversible. So commitment here at this particular stage, that is at the determination stage, it becomes irreversible. The cell, it cannot revert back. Then comes that the loss of the competence for the alternative fates or the positive feedback loop or the chromatin remodeling. So the chromatin remodeling basically takes place here, and which majorly plays a role that once the gene are being expressing to form a particular kind of cells, then those kind of cells only be produced, right? So what happens in the chromatin remodeling, what happens is like a particular part of the DNA only is there, which would be expressing itself. So that is why those cells which has to produce the muscle cells, they only produce the muscle cells. They do not produce the neural cells, because the genes which are expressing, they are basically for the production of muscle cells. They are not for the production of the neural cells. So this is how the determination basically takes place. Now comes the differentiation. This is yet another stage at which the cell is more restricted, right? Now the pathway is being decided, the cell is being following pathway, and now it is restricted to become a certain type of cell type only. So what happens here? The changes involved in the progressive diversification of the structure and the function of the cells, right? Now since the cell has to form a particular type of cell fate it has to achieve, then what happens, internally there is a certain kind of modification which takes place at this particular stage, right? So differentiation process is basically when the cell from an unspecified stage, it gets differentiated into a specified form, right? So from the Blastula stage, which is an unspecified stage, a cell gets differentiated and it gets converted into a more specified stage, that is the neural cells it will form, right? So here comes the role of differentiation. Then other statement, it says that the acquisition of the characteristics that allow the different cell types to perform their functions, right? There will be different cell characteristics to allow the different cell types to perform cell functions, right? So there will be different characteristics which would be produced by the cell that will lead to the formation of a particular cell type only. So their functions will decide which kind of cell it will be producing, right? The development of the specified cell types through different gene expression. So gene expression here plays a role. So once the gene it is expressing for the production of the muscle cell, it will only produce the muscle cell. So all of these things plays a role here. The proteins, its functions plays a role, right? Since the cell has already decided that which fate it will be achieving, for that the proteins will will be producing, and the genes will be expressing accordingly, and after that the expression will take place likewise, right? A cell is said to be differentiated when it shows the specific characteristics. So from the unspecified cells, the cells will start showing certain specific characteristics which are basically known for that particular type of cell type only, right? Then comes the generally differentiation is irreversible. So differentiation is also something which is irreversible, right? But under in vitro conditions, they can be differentiated given some specific gene signals. So if some specific signals is being given and they are being um, or incubated into like outside in vitro condition, then if certain signals are being provided to them, they can revert back. So at that particular point of time, the reversibility they can show, but normally they don't show the reversibility, right? So this is the entire differences between all these three terms. So the major pathway of development, what is the major pathway? So this order you need to remember is firstly the undifferentiated cells. So I'll write it here. Maybe it's not everybody is able to see. So undifferentiated cells comes, right? Undifferentiated cell comes, then it produces, then specification takes place. Right, specification or commitment takes place. The cell it decides what particular fate it will be achieving. Right, after that comes the determination stage. So cell gets determined. So here cell gets determined that to which particular pathway it would be following and which particular cell fate it would be achieving. Right, then comes the differentiation. So this is the order in which you need to remember the particular pathway, because questions come in CSIR NET exam related to this pathway. What is the order? So in jumbled up way they'll be giving, and it will be very confusing at times to like mark the right answer. So cell fate determination. So here is an experiment where you can see that in A part what happens. So here is the blastula. So there would be in colored form, you can say that see that there would be red colored cells and there could be yellow colored cells, right? So these red color cells, they are specified to produce the muscle cells, and the yellow color cells, they are specified to produce the neurons. So when these particular cells, they are being isolated and they are being grown in isolated conditions, separately they are being grown, then what happens, this red cells, they basically produces the muscle cells, and the yellow cells, they basically produces the neurons. But in another diagram, it is being seen when both of these cells, like the red cells which were producing the muscle cells, when they are being placed into the in between the yellow cells, that is the neuron producing cells, then what happens is these cells and the external factors are also being provided here. The factors which are responsible to produce the muscle cells, these are also being given here. Then what happens, the firstly, the specification is labile. Right, it can revert back. So yellow cells, so you can see here that the muscle cells that we were growing inside the cells which were producing the neurons, and the external factors that we were giving it to was the neuron producing cells. Then what happens, the muscle cells also produces the neuron. Right, here also it is producing the neurons. So there is the reversibility. You can see that the cell can revert back. So it depends upon the external factors. What kind of external factor you are giving, at that basis also the particular kind of cell, it can revert its cell fate. Right. Then comes in C portion. In part C, it says that the specification is irreversible. So at this particular point of time, the cell is in its determination state. Right. This is the determination stage. And earlier it was basically the specification or the commitment stage, right? Or commitment, you can say. So interrelating both these terms is very much important with respect to the diagram, because questions are majorly being asked from the experiments also, right? So in part C, you can see that the particular cell is in its determination stage, and it has determined which kind of cell it will be producing. So even after the external factors are being given to the cell, and both of these cells are being grown together, then what happens, the specified muscle cells is determined and becomes the muscle despite of the surrounding environment. So even after the surrounding environment is being changed and the factors that produce the neurons that is being provided, then also the muscle cell, it will only produce the muscle cell type only. It will get differentiated into the muscle cell type only. It won't get differentiated into the neurons. So here the cell is in a determination stage, and here in part B, the cell in its commitment stage, right? The cell has yet not decided which fate it will be producing. it will be achieving, sorry. So here is a question for you, right? So first question you need not to attempt, because this part we have not yet discussed. So second is the second question you need to attempt. This question we have like discussed already. So this question you need to read out, and you need to understand what this question is asking, and you need to give me an answer for this, right? Okay, Jagdish says, once again, I need to explain the experiment number B. So in experiment number B, what is actually happening here is, no, so this is the blastula, right? There are red colored cells and there are yellow colored cells. Yellow colored cells basically produce the neurons, and the red color cells basically produce the muscle cells. Now, when one of these cell is being isolated and it is being placed inside that environment where the neural cells are basically like they are developing, and the external factors that we are giving into that petri dish is basically for the production of the neural cells, then what happens, this particular muscle cells, it gets differentiated into the neurons, right? So neurons it will be producing, right? So neurons it would be producing, the neural cells it would be producing. So at this particular point of time, we can say that this cell is majorly in its commitment state, right? So specification here is labile. It can revert back. So it is a reversible process. Because what is happening here is that on the basis of the change in the external environment, the cell is changing its fate also. It is converting into a different type of cell, right? Can we say that a cell, when changes its fate, its environment condition get repressed and other cell environment condition dominate? Yes, absolutely, Mahima. So this is what happens here. It get influenced by the external factor. So at this particular point of time, the cell is in its committed state, right? The specification is labile here, it is reversible. While coming to the C part, the specification is irreversible. The cell is in its determinate state, right? So I hope I have given the answer. That Jagdish, are you able to understand now? Right. So here is a question. So in the second question, you have to majorly attempt this particular question. You need to give me the answer for this. Right. So the question says that the blastomeres of the two-cell embryo, right? Here the two-cell embryo blastomere is basically being taken and are separated, and they are being basically being allowed to develop independently, right? So independently they are developing. Each blastomere gives rise to young ones, right? So each blastomere gives rise to the young one. The type of development pattern suggests that the potential of the development is, like, which one option would be the correct one? It would be the greater than the fate, less than the fate, equal to its fate, or the independent of fate, right? So I would be expecting, I would be inviting the answers from all of you. Okay, Kitana says answer is B, less than the fate, and Ankita says answer is C. Okay. So were you able to understand what the question is actually asking? So for this, the option C is the correct one, equal to its phase. Here basically the question is majorly asking about the potential of the development. Here the potency plays a role. right? The question is being asked in such a term where you need to like figure out what it is majorly being asking. So from potential, you need to understand it is basically asking about the potency. So potency we have like read earlier in the slides, which says that the potency is always equal to, or it will always be greater than the cell fate, right? Mostly it is equal to the cell fate. So blastomere of the two embryo are separated and allowed to develop independently. So independently they are developing, right? So whatever cell we have picked from the blastomere, both of these cells, they are capable of forming independently. They are capable of forming each type of cell, right? So their potency is actually equal. Whatever this cell can produce, the other cell can also produce the similar type of cell. So because of this, like blastomere, what happens in blastomere, the cells at the two cell stage. So here you need to remember and like observe which cell stage it is majorly being asked. Question is majorly asking. So it is the two cell stage. At two cell stage, the cell is majorly in its totipotent form, right? A totipotent cell is capable of forming any type of cell, like it is capable of forming the whole organism itself. So therefore what happens is both the individual cells, they are capable of forming each type of cell. So their potential, their potency is same. So it would be equal to its fate, right? This would be the correct option. Option C is the correct one. So now comes the autonomous specification, conditional specification, and synthetal specification. Now the specification is further being divided into three major categories. That is, autonomous specification is there, conditional specification is there, and synthetal specification is there, right? So in autonomous specification, what happens is specification by differential acquisition of different cytoplasmic molecule present inside the egg. Right. So here plays the role of certain cytoplasmic determinants would be there. Right. These will play the role and it will decide like the particular fate of the cell. Right. So it says that invariant cleavage produces the same lineages in each embryo of the species. Blastomere fates are being generally invariant. So fates of the blastomere is invariant. Right. And the cell here, basically what happens, this and the thing which you need to remember here is that the cell potency is actually equal to the cell fate. So whatever the fate of the cell would be there, whatever kind of cell type it would be forming, it would be equal to its potency. Then it says that the specification is through intrinsic signal from cell's own cytoplasmic determinants. So intrinsic factor plays here the role, right? So these intrinsic factors are majorly responsible, or it influences the autonomous specification. So these intrinsic factors, what happens, are present majorly in the form of cytoplasmic determinants inside the cell, inside the blastomere. So these, the concentration of these cytoplasmic determinants will majorly decide which part of the cell it will be forming, right? The specification of the cell would be decided like from by the cytoplasmic gradients. It's a great cytoplasmic determines its concentration inside the cell. What is its concentration? And cell type specification precedes any large scale embryonic cell migration. Then it says that the produces mosaic. So here the mosaic formation takes place. In autonomous specification, what happens? A mosaic formation takes place. How mosaic formation takes place? That if a blastomere, so suppose we took a blastomere and we like took out a certain kind of cells from the blastomere, and rest of the cells are remaining there. Then what happens is those cells which are being removed, because of them, the cells, like they were basically be designed to produce a certain kind of cells, no? So they are being removed. Then what happens? A mosaic is being formed. Mosaic it is like somewhere the genes would be expressing those kind of cells would be formed, and somewhere it won't be formed. So in form of patches, we can see that the blastomere, there are the patches is being formed inside the blastomere, where certain types of cells which are present, it is producing, it's causing its effect, while the certain types of cells which is missing, there is, we cannot see any kind of cell formation, right? So it says that the produces mosaic, determinative and development, and cells cannot change fate if the blastomere is lost. So it cannot reach or achieve its fate if the blastomere is lost, if those cells are being lost, So those cells are very much important. Unless until those cells are present, the cell cannot achieve its particular fate. Then comes the characteristic of, it is the characteristic of most of the vertebrates. So autonomous specification is the characteristic of most of the vertebrates, right? Then comes the conditional specification. So here it says the specification by interaction between cells, relative positions are important. So the certain types of cells, which the positioning of the cells is very much important here, and those the positioning of the cells basically decides that which particular part of the cell this it would be forming, right? And interactions, it interacts with the environment, and it says that the variable cleavages produce to no variant fate assignments to the cells. And here the cell potency is greater than or equal to its fate, right? So the potential of the cell is greater than or equal to its fate. So whatever the fate the cell want to achieve, its potential would always be greater than or equal to its fate in case of conditional specification. Then it says, while in case of autonomous specification, what did we study? We studied that the cell potency is equal to the cell fate. Then comes the specification is through the extrinsic signal from the cell's environment. So here the extrinsic signals plays a role, right? If the extrinsic signals, or we change the external signals, then the cell's fate, it gets reversed. Like it can like produce another type of cells. So this conditional specification is basically an example of the commitment, or the specific commitment that we discussed earlier, right? And then what happens that the massive cell rearrangements and migrations proceed or accompany specification. So here like massive rearrangement of or the migration of the cells takes place, right? And on that basis only the particular pattern is being formed in the cell, what particular type of cell it will be forming. Then comes the capacity for the regulative development, allows the cells to acquire different function, right? So regulative development takes place in form of regulation, like certain cells has to be formed, certain cells has not to be formed. So the capacity of the cell for the regulative development, it allows the cells to acquire different functions. So different functions, the different cells would be forming different functions, and when those functions are being conducted, on that basis a particular cell type is being decided, right? Then comes the characteristic of all vertebrates. It is the characteristic of all vertebrates, and it is not found in invertebrates, right? Then comes the synthetal specification. Synthetal specification is the property of majorly insects. So in Drosophila melanogaster is the most important example for this, for synthetal specification, right? So what happens in here is the specification of the body region by interactions between the cytoplasmic regions prior to the cellularization to the blastoderm. So what happens here is, as we can see, in case of Drosophila, what happens? Yeah. In case of Drosophila, what happens is there are Drosophila has multi-nucleuses, right? Multi-nucleated structures are being formed, but the cell is single cell. Inside a single cell, there is multi-nucleated multi-nucleuses which are being formed, and it is majorly being observed in Drosophila melanogaster. So insects have this property of synthetal specification. So it says that the variable cleavage produces no rigid cell fates for the particular nuclei. So these nuclei, they are capable of forming any kind of cell, right? Any part of actually the part of the Drosophila. So it is not restricted to produce some like definitive part. It is able to produce any part of the Drosophila, right? So here no restriction comes here. And then comes that the cell potency is always greater than or equal to fate. So here also the potency of the cell is greater than or equal to its fate. So the potential of the cell to form any type of a cell, it is more as compared to the cell's particular fate, the cell to which particular type of cell it is committed to, right? Then comes the specification is through intrinsic signals by a gradient of cells' own morphogens, right? So there is the morphogens present inside the cells. These morphogens are basically present intrinsically, and these morphogens are majorly responsible for the specification of the cell, right? So here also intrinsic factor plays a role. In autonomous specification, intrinsic factors were influencing the cell fate. In conditional specification, external factors were influencing the cell fate. And in syncytial, the intrinsic factors are responsible, or they are influencing the cell fate, right? Then comes that the specification happens during the syncytial phase of the embryo development, where the nucleus gets specified, right? So when the nucleus gets specified to produce a particular cell, then only the nucleus gets specified to produce a particular cell. So here it says that the after cellularization conditional specification is most often seen, right? When the cellularization takes place, when these nucleuses they are being separated by production of certain cells, so in between their cells would be formed and these nucleus will get separated, then only the cellularization will take place, right? These cells will produce different types of different parts of the organism. So conditional specification only takes place when the cellularization of the nucleus takes place, right? Then comes the characteristic of most of the insect classes. So I have given example here. The Drosophila melanogaster is major example for syncytial specification. So on that basis also there is a question here. So it says that during embryogenesis, differentiation of a cell involves process like specification and determination. In this regard, which of the following statements are correct? So question says that during embryogenesis, differentiation of the cell involves processes like specification and determination, right? So specification and determination process takes place. And in this regard, which of the following statement is correct? So autonomous specification leads to mosaic development. Second says the autonomous specification leads to regulative development. Third says that the conditional specification leads to regulative development, and the conditional specification leads to mosaic development. So which of the following options is correct here? So now only we studied about the autonomous specification, which says that the autonomous specification leads to mosaic formation. So mosaic development takes place actually here. In autonomous specification, mosaic development takes place, right? And apart from first option, the third option, it says that the conditional specification leads to regulative development. So option 3 and option 1 is the correct here, and option A is the correct option here, right? So conditional specification, we studied about the regulative development, right? That what happens in conditional specification, even if the extrinsic factors, if they are being changed, then the cell has the potency, or cell has the capability to revert back, right? So regulative development basically means with that only. So conditional specification leads to regulative development. So both of these options are correct here, right? Okay. Second option says that second question says the ability of the cells to achieve their respective fate by themselves without the influence of the neighboring cells is called. This kind of specification, what it is being called? Like it is not influenced by the external factors or the influence of the neighboring factor. So which particular specification will being considered for this question? First says that the autonomous specification, second says the conditional specification, third says the syncytial specification, and last one says the non-conditional specification. So which particular option would be correct here? Okay, Manish says A, Jagdish says A. Okay, most of the people are saying A option, right? So this is the correct option, autonomous specification, where basically it doesn't get affected by the external factors. Intrinsic properties are majorly responsible for autonomous kind of specification, right? So coming to the next slide. So yeah, coming to the next slide, it says that the example of the autonomous specification has been given here. So autonomous specification example is being given here. So we have taken example of the tunicate embryo. So what happens in here is that the 4 blastomere pairs of the 8 cells embryos dissociated. So here what happens, this is the blastomere we have taken, right? 4 type of cells are there, right? So the first one, I'll denote it like first, second, and third, and fourth, right? So the first one is forming the nerve system, and the third cell is forming the, either it can form the endoderm or the notochord, and the second cell it is forming the ectoderm, while the last cell it is forming the muscle cell or either mesenchymal cell. So when these particular cells inside the blastomere, they were forming these kind of different cells, right? In a normal condition, when these different cells, they are being isolated, right, and they are being grown at different conditions, then what happens is the second cell, we can see that the second cell, it forms the ectoderm only. So whatever it has formed inside the blastomere, that very cell it is forming the outside condition in the isolated form. And the fourth cell is also forming the same thing, right, muscle and the mesenchymal cells. And the third cell is also forming the same thing, notochord. But the second cell is the one which is forming the ectoderm, right? Its cell fate has been like changed. So what is it basically telling here? So it says that the 4 blastomere pairs of the 8 cell embryo is being dissociated, right? Secondly, all forms of the structure it would have formed had it remained in the embryo. So all the different cells, they are forming the same structure only when they are being isolated, whatever structure they were forming when they were inside the blastomere. But tunicate nervous system is conditionally specified, right? So this cell is basically conditionally specified, right? So it depends upon the external factors. When the external factors are being changed, then what happens, this particular cell, it instead of producing the normal like nervous system, it produces the ectoderm. So this is what actually happening here, right? So this particular cell goes into the conditional specification, while rest of the forming the same thing. So these are the in the autonomous specification state, while this one is the conditional specification stage. So did you understand this particular example? Another important the experiment that was being proposed by the rocks experiment, it was, and it says that when there was a fertilized egg, it was in its cleavage state. So hot needle was being poked inside this fertilized egg, and it was basically in the 2 cell stage at that particular point of time. Then what happens is that certain like half of this 2 cell stage, this particular whole cell, half of that cell is destroyed. It is dead tissues are being formed there, and half of the cells, they are producing the live tissues, right? So half is being destroyed and half is being forming embryo in the Neurula stage. So result here was that the frog embryo was in a like mosaic of self-differentiating, right? So mosaic is being formed here. So those cells which were responsible to produce a certain type of cells in this half part, they were being removed. When they were being removed, there was like tissues got destroyed, and they produced the dead tissues. So it is being like, from this the conclusion can be made that those cells were required to produce a certain kind of cells. When these are being removed, the particular part of the blastomere or the egg, it would be destroyed, right? So mosaic of self-differentiating parts is here, and each cell received a set of nuclear determinants and differentiated accordingly. So nuclear determinants each cell received, and on that basis only they differentiated themselves. Then coming to the example of conditional specification. So for conditional specification, here it is given an example. So it was being like a blastula was being taken here, right? So the back cells, the cells which were present in, like which were majorly responsible for producing the back of the organism, from there the cells were being taken, and they were being isolated, or they were being kept inside the belly part, right? The part which were forming the belly, it was being isolated, and it was being put there. Then what happens is, it can be seen that no transplantation, normal development takes place at that particular point of time. Like so these cells which were being taken from the back to the belly, they were forming the belly tissues only, right? On the basis of the external factors, it is getting changed, right? It's fate getting changed. Then coming to the B part. So in B part, what happens is when the glass needle, it removed certain cells. So cells were being actually removed from here. They were not being placed into some other place, but they were being removed. When they were being removed, then what happened? Then also the normal development is taking place, right? So this is the conditional specification. So conditional specification basically depends upon the external factors. If the external factors are being changed, then the cell fate also gets changed, right? So basically the interaction with the neighboring cells takes place, and on that basis only the cell fate is being decided. And what happens here, the very good example of this is in the 4-cell stage of the plastomere. What happens? We have removed one cell. So one cell is being removed. When one cell is being removed, the other cells, they are capable of compensating the effect of that removed cell. While in case of autonomous specification, it was not the case. When a particular type of cell was being removed, the cells were basically destroyed. The other cells, they were not able to compensate its requirement, right? While in case of a conditional specification, the other cells are capable of compensating the property of the cell which is being removed, and they are able to produce a particular cell type, right? Normal cell type. Then comes the example of the synthetal specification. So in synthetal specification, as I have told you earlier also, it is the property of the insects basically. So Drosophila is one of the great example for synthetal specification. So here says that the nucleus cycles during Drosophila early development takes place. So here is this cycles consisting of 4 to 6 nucleus is being produced. So firstly, the nuclear division takes place, right? Then only the cellular division takes place here. So at 30-minute, fusion of the sperm and the eggs and nuclei takes place, right? So what happens? Here is the nucleus inside the egg, and the cytoplasm of the egg, it consists of all the proteins which are required for the development. And then what happens? Fusion has taken place. Here the sperm has come and it has fused with the nucleus. Right, the nucleus has entered here and the two nucleus have fused. Then what happens? After 70 minutes, it was being noticed that the nuclear division creating the syncytium was being observed. Right, different nucleuses were being formed. So syncytium is basically the formation of different nucleuses. Right, so different types of nuclear division basically takes place here. After 70 minutes, it was being observed. After 90 minutes, it was being observed that the nuclei migration takes place. Right, so this nucleus, it migrates into the periphery. So here also in the diagrams, it is being seen here that after 90 minutes, the nucleus, it gets migrated to the periphery, right? Then after 2 hours, it is being seen that the cellularization takes place, right? In cellularization, what happens? These nucleuses, they are being separated from each other, and cells are being produced surrounding this nucleus, right? Then after 3 hours, what happens? Cellular blastoderm is being formed. So all of these nucleuses, they are able to produce, they have committed themselves to produce a certain cell type. So inside the Drosophila, it will produce a certain cell type. After 3 hours, it is being seen that the cellularization takes place, right? This is the example of syncytial specification in Drosophila. So this is also being internal factors are majorly responsible for this type of specification. And here also the potency is greater than and equal to the cell fate. Right. Then comes the morphogen and the cytoplasmic determinants. So you need to know what is the difference between both of these terms. So for morphogens, it says that it is a signal that affects the different cellular responses at different concentrations, right? So morphogens are basically the signals. When they are present at different concentrations, their concentration differentiation basically decides the responses that would be produced for the cell to produce a particular type of function and get converted into a different type of cell, right? Then it says that it specifies more than one cell type by forming a concentration gradient, right? Due to its concentration gradient only, it can form multiple types of cells, right? Wherever the concentration is high, it will form a certain kind of cell. Wherever the concentration is low, it will form a certain kind of cell. And wherever the concentration is moderate, it will form a certain kind of cell, right? Cells respond to different threshold concentration of the morphogen by activating expression of the distinct sets of genes, right? So there are sets of genes which are being expressed due to this concentration gradient which is basically being responsible to produce a certain kind of response inside the cell. As a result, different parts of the cell is being formed. Coming to the cytoplasmic determinants are basically the molecules. These are the molecules, while morphogens were the signals. So these molecules that are differently that they are differentially inherited from the mother cells. So these cytoplasmic determinants are majorly present in the egg itself. It is already present in the egg and in an asymmetric manner, right? It specifies only one type of the cell. It specifies just one type of cell, right? While morphogens they were specifying different types of cells because of their concentration gradients, depending upon what type of determinant has been acquired. So other thing, it says that the cell activates an intrinsic signaling cascade by the differential acquisition of the determinants. So when these determinants and they respond, or they are being produced, a cascade of events takes place, and the cell activity, basically the response and the cell activity basically takes place due to these cascade of events taking place due to the cytoplasmic determinants, right? So this is the general comparison between both morphogens and cytoplasmic determinants. Since we are like the time is we have crossed the time actually. So what happens now? You can actually note down all these things that are given on in the table itself, and we'll be proceeding with the other unit, that is the unit 6. I'll be covering on Monday, right? So here we come to an end of the session because since the time is very much and I have exceeded the time, so we'll be ending up the session here only. So now I would want to tell you that there would be certain sample papers related to this particular chapter. Whatever you have understood till now from the session, you can attempt those questions from there, right? So download a link is being given in the description, so you can go through that. And quick reminder that at 5 p.m. we'll be conducting a webinar session where we'll be discussing about the paper discussion will take place, basically, right? The difficulty level of the paper, the overall analysis of the paper take place. The whole paper discussion, question by question, it will be discussed from the next week. So stay tuned and like be there at that particular point of time, so you'll get an idea, right? So thank you very much.